Okay, welcome back, everyone. We just finished talking about the endosymbiosis, which is kind of crazy, and I do hope you read your textbook to kind of explore this theory a little bit more. This is something that I do expect you to be able to describe to me, both how we know, like what evidence there is that supports it, that inside out and outside in theories are going on. Next, let's explore some more of our eukaryotic cells by exploring the endomembrane system. So the endomembrane system is kind of this continuous layer of membranes going from the nucleus all the way to the cell membrane. Within the endomembrane system, these are structures that are going to create proteins that are going to go to various organelles, that are going to go to cellular membranes, or going to be secreted from the cell. Anything your cell needs to secrete and send to another cell is going through the endomembrane system. These proteins are packaged and directed to various destinations using vesicles. So we're also going to talk about vesicles within our endomembrane system. Within our endomembrane system, once we branch off from the nucleus, the first part is the endoplasmic reticulum. Almost every eukaryotic cell has this network of membranes. You can see it's just layers of membranes that are connected to each other. And these membranes are within the cytoplasm, and they're called the endoplasmic reticulum. These have tubular membranes and flattened sacs called cisternae. And within the middle of them is the lumen. Hopefully that's a term you've heard before. Lumen is used to describe the interior of a lot of different cavities. And the ER is continuous with all the other membranes in the cell. The endoplasmic reticulum can be rough or smooth in appearance, so whether it has ribosomes attached to it or not. The rough ER is rough because it has all those little bumpy ribosomes all over it. So these little studs of ribosomes all over it. In this picture, you can see kind of these little dots all over it. These ribosomes are important for synthesizing polypeptides that are going to accumulate within the membrane, inside the membrane, or be transported across the membrane. And free ribosomes are not associated with the endoplasmic reticulum. However, ribosomes, whether they are free or bound, are identical to each other. A free ribosome can attach, and a bound ribosome can detach and become free. They're structurally the same. The only difference is if they are attached or not attached. The smooth endoplasmic reticulum does not have a role in protein synthesis. It does not have ribosomes on it, so it's not making any proteins. Instead, what the smooth ER is doing, it's making lipids, things like steroids, cholesterol. It is inactivating and detoxifying things that are potentially harmful. So, if you are out celebrating New Year's Eve, the next morning your smooth ER is going to be very active, detoxifying that champagne. And the sarcoplasmic reticulum is a type of endoplasmic reticulum that is important in contraction. You'll talk about sarcoplasmic reticulum like in muscle cells. After the endoplasmic reticulum, we move on to the Golgi apparatus. The Golgi apparatus is usually very closely related to the endoplasmic reticulum, both in location and in function. And it'll be a series of flattened vesicles known as cisternae. And it is important in processing and packaging secretory vesicles. So these vesicles come from the endoplasmic reticulum, then come to the Golgi where they are received. And those proteins will be processed and then sorted out to the different places where they're going. Then Golgi also is responsible for synthesizing some complex polysaccharides. And it takes transition vesicles that are coming from the endoplasmic reticulum right into the Golgi. The vesicle contents from the ER are going to be modified and processed within the Golgi complex. We're going to talk about this a lot. I think it's chapter 17 when we get into talking about our endoplasmic reticulum and Golgi and all the ways that these proteins can be modified. One example would be secretory and membrane proteins are going to be glycosylated within the Golgi. That means that short carbohydrates are going to be added to those proteins. This starts within the ER, but is completed within the Golgi. Then when the substances are fully processed, they can move from the Golgi to other locations. These little vesicles are going to bud off of the Golgi, and these vesicles are then able to move on. Maybe they're going to fuse with a lysosome or create a lysosome. Maybe they're going to fuse with the plasma membrane or release contents out of the cell. Once materials are processed by the Golgi complex, materials that need to be exported from the cell are going to pass into these vesicles that move to the plasma membrane, and you can see this membrane of the vesicle fuses with the membrane of the plasma membrane, which allows the materials within the vesicle to be released. This is exocytosis, exocytosis, materials exiting from the cell. So the endoplasmic reticulum, the Golgi, secretory vesicles, and lysosomes make up the endomembrane system of the cell. And this is responsible for cell trafficking. And proteins always move in this one direction. They're going to move from endoplasmic reticulum to Golgi to vesicles to outside of the cell or fuse with the cell membrane to create these functional molecules. We mentioned lysosomes there. Lysosomes are single membrane organelles that have these important enzymes called hydrolases in them. Hydrolases are able to digest biological molecules. So if you have an old worn out protein, you have an old worn out organelle, you can send them into that lysosome. And these hydrolases are able to break them down. But hydrolases aren't going to be just floating around the cell breaking things down. It's really important that we keep them sequestered within the lysosome so the functional things are still functioning without being broken down, and things that need to be broken down can be sent to the lysosome. The inner lysosome does have a special carbohydrate coating on it that helps protect the lysosome itself from being digested. Next are peroxisomes. Peroxisomes are pretty similar to lysosomes in terms of their size and their appearance. They are also surrounded by one membrane, one bilayer, and they can perform some different functions depending on what type of cell they're in. They're especially prominent in humans in liver and kidney cells. Peroxisomes are named after hydrogen peroxide. They create hydrogen peroxide. H2O2 or hydrogen peroxide is very toxic, but it can be formed into water and oxygen by an enzyme called catalase. And eukaryotic cells have processes to create H2O2, and these reactions are confined within the peroxisomes or catalases so that cells will be protected from being harmed by the peroxide. Peroxisomes are able to detoxify things if there are harmful compounds. They can also catabolize some unusual substances. In animals, they're important in oxidative breakdown of fatty acids. It means fatty acids are going to be broken down, especially if they're longer fatty acids. And some human diseases can result from defects in peroxisomal enzymes that are involved in breaking down those fatty acid chains. In plants, peroxisomes during seed germination, specialized peroxisomes called glyoxysomes are important in taking the stored fat in that seed to turn it into carbohydrates for the seed to have energy to start sprouting. And leaf peroxisomes are important in photosynthetic tissues because they're involved in photorespiration. That is light-dependent uptake of oxygen. So photorespiration is allowing that plant to use the energy of light to uptake oxygen and release carbon dioxide. So that's not photosynthesis, right? That's the opposite of what happens in photosynthesis. Photorespiration. Some cells have a vacuole, which is a membrane-bound area used for different functions in different cells. In animal and yeast cells, vacuoles can be used for temporary storage or for transport. After phagocytosis, a cell that has just phagocytosed brought a food particle in will have this vesicle this membrane-bound particle called a phagosome. And when the phagosome fuses with the lysosome, it allows the lysosomal digestive enzymes to break down whatever was just phagocytosed so that that food can be providing nutrients to the cell. In mature plant cells, they typically have one very large vacuole called a central vacuole. You can see in this micrograph, it's pretty big, right? The vacuoles are really large. It can take up to 90% of the space in that cell. The main function of a central vacuole in a plant is to maintain turgor pressure. So because it's filled with water, it's kind of providing pressure against the cell wall and helps keep that plant from wilting. If that vacuole starts to lose water, it no longer is able to press against the cell wall. It won't provide enough pressure, and that's what causes those plant tissues to wilt. Next, we have ribosomes. Ribosomes are responsible for making proteins. So ribosomes are the site of protein synthesis. All proteins are made at ribosomes using ribosomes. There are many more ribosomes than almost any other intracellular structure because it's just so important to be creating the proteins that your cell needs. They are found in all cells. We did discuss how ribosomes are slightly different in our different types of cells, bacterial cells, archaea cells, and eukaryotic cells, which allows that to be a target for antibiotics. And each different cell type has its own unique type of ribosomal RNA. The ribosomes are made of RNA and proteins. And so in these different types of ribosomes, we have slightly different ribosomal RNA. Ribosomes are more numerous than other cell structures. So if you were to count a prokaryotic cell, you could count thousands of ribosomes. And a eukaryote, you may count millions of ribosomes in it. Ribosomes in mitochondria and chloroplasts are similar to bacterial ribosomes. And this is especially true if you start looking at the nucleotide sequences of the ribosomal RNAs. Ribosomes have two subunits, a large subunit, which is a lighter green, and a small subunit, which is a darker green. And they have different sedimentation coefficients. This is math that does not math, and that's okay. And actually, let me pause here. Okay, I just saw a typo, so I just corrected it, so your screen may have changed a little bit. So this is math that doesn't math, because we are not just adding like weight. We are looking at a sedimentation coefficient. So this means that if you like centrifuged a cell component, what layer you would find these different things in. So if you were to centrifuge this, the large subunit by itself would be in the 60S layer, the small unit by itself would be in the 40S unit. If you were to centrifuge it together, it would be in the 80S layer. And those do not math, and that is okay. They don't add up because we are looking at a combination of not only its weight, but also its shape and its size. So those are the large subunit, small subunit, and combined subunit. In bacteria and archaea, the large subunit has a sedimentation coefficient of 50S. The small subunit is 30S, and together it's a 70S. So again, it does not add. That is okay because we're looking at where it would centrifuge because it's a function of not only its mass, but also its shape. So here, that tells you about that if you want to read about that some more. These numbers are correct: 60, 40, 80, 50, 30, 70. Then let's talk about the cytoskeleton. The cytoplasm of eukaryotic cells has this intricate, organized, three-dimensional array of interconnected proteinaceous structures. That is a mouthful. That just means there's a bunch of proteins that forms this webby skeleton, and that's called the cytoskeleton. Just like your bony skeleton in your body helps organize and give your body its shape, the cytoskeleton helps a cell have its distinctive shape and internal organization. So here, just by coloring the cytoskeleton of the cell, these lit up parts are the different cytoskeleton. You can see what the shape of that cell is going to be without seeing its plasma membrane or anything. Cytoskeleton is also important in cell movement and cell division. It lets things move, so like it can form a pseudopod to stretch and move, or it can create flagella that allow the cell to move, and to allow the cell to divide, to pull DNA to pull the nucleus to opposite sides of the cell, to allow the cell to divide in half. Proteins related to eukaryotic cytoskeleton have been discovered in bacteria and archaea, and also seem to have a role in maintaining cell shape. The cytoskeleton is a framework for positioning and moving organelles and macromolecules within the cell. It may also help move ribosomes and enzymes. And even about a third of the water within a cell is bound onto the microfilaments and microtubules. There are 3 different structures of the cytoskeleton: microtubules, microfilaments, and intermediate filaments. We're going to come back in later this semester to actually explore these different structures in a little bit more detail. For now, you can just know that those are the structures that are there. Okay, so now we have talked about all of what's inside of the cell. In our next video, we're going to come back and talk about what's kind of going on the outside of the cell. Its cell wall or extracellular matrix. I'll see you then. Bye.